SLM, welcome to our first STEM day of the 2021 school year. Today's STEM challenge will involve something that is very commonly found this time of year: pumpkins. We will be completing the STEM challenge. How many pumpkin seeds can I stack on top of one another? You will be doing an experiment involving soaked pumpkin seeds as well as dried pumpkin seeds. You will need for this experiment your STEM activity guide provided to you by your teacher. You will need one pumpkin. You will have to cut it open and pull out the seeds. You will need a bowl to soak your pumpkin seeds. You will need an oven-safe tray to dry your pumpkin seeds. You can also place the pumpkin seeds in the sun to dry for several hours if you do not want to use an oven. And you will need a surface to stack your pumpkin seeds as well as a ruler to gather data. First thing that you need to do is think about how many seeds can you stack successfully without the stack falling over. You are to complete your hypothesis, which is the first step on your STEM activity guide. I will be able to stack how many seeds? How many seeds do you think you'll be able to stack? Do you think dried seeds or do you think soaked seeds will make a better stack? How many will you be able to stack? Which one will be a taller stack? Will it be the dried seeds or the soaked seeds, the wet seeds? So you will fill in your hypothesis before you proceed on. If you look at your materials, you again will need one pumpkin, one bowl, several seeds from the pumpkin, a ruler, a tray, an oven-safe tray to soak your seeds. You will open the pumpkin and take out your pumpkin seeds, soak half of them, and dry the other half. Once you have your dried and your soaked pumpkin seeds, you will attempt to stack them one on top of another, flat side down, right? You're not stacking them like this, you're stacking them flat side down. You will place the pumpkin seeds one on top of the other very carefully until you create a stack. It might take some practice to do this step. Once you have created your stack that is successfully standing and not falling over, you will count the number of pumpkin seeds in your stack and record it in your data table that is located under the results section of your STEM activity guide. You will also use your ruler, and remember to use the centimeter side, to measure your stack. Once you have measured your stack, you will again record that data in your data table. Once you have recorded your data, you will answer the question: were you successful? Were you successful in completing this challenge? Were you able to stack pumpkin seeds, either the dried ones, the soaked ones, or both? Then you will complete a graph, a bar graph, with your data from your data table. Well, students, were you successful at creating your stack of pumpkin seeds? Which one worked better? Did the wet seeds work better, or did the dried seeds work better? Make sure you let us know your results in the comment section below. Now we're going to move on to our connection to life science today. Our connection to life science is that of a pumpkin, right? We're looking at the seeds of a pumpkin. So we're looking at the structure of a seed. What is the purpose of a seed? What is the structure of a seed? Pumpkins come from what we call flowering plants, right? Pumpkins are a green vine with beautiful yellow blooms. If you've ever grown your own pumpkins, you have seen the vine of a pumpkin plant as well as those beautiful yellow blooms. All flowering plants are what we call angiosperms. The term angiosperm means flowering plant. So any plant that produces a bloom, produces flowers, is called an angiosperm. If plants do not produce a bloom, like a pine tree or other conifers, they're called gymnosperms. They do not have flowers. They do not produce flowers. Angiosperms all have the fact that they produce seeds in common, right? So angiosperms produce the flower is pollinated, and the seeds are grown inside a fruit. So most angiosperms have a fruiting body of some kind, right? Pumpkins are technically the fruit of a pumpkin plant. Inside the pumpkin are bunches and bunches of seeds. What is the function of a seed? What is the purpose of a seed? If you guessed that a seed is a baby plant, you would be correct. A pumpkin seed, right, the little white pumpkin seed that you pull out of a pumpkin, has multiple parts. It's not just a seed that you put in the ground to create a baby plant. The pumpkin seed has a seed coat. So the white part that you see when you pull a pumpkin seed out of the pumpkin is called the seed coat. Sometimes we call that a husk. Inside that husk are multiple parts. One of those parts is called a cotyledon. Pumpkin plants actually have two cotyledons. Pumpkin plants are called dicots, meaning they have two cotyledons. There are also some flowering plants that have only one cotyledon. These are called monocots. What is a cotyledon? Well, the function of that is to provide the food source necessary for the baby plant, which is called the embryo. In monocots, you only have one cotyledon, one food source. In a dicot, you have two. Now, the cotyledon will provide the food for the embryo, but it's also what will become the first leaves of the sprout. So in a dicot, you actually have two leaves coming from the seed. In a monocot, you only have one leaf coming from the seed when it sprouts. When you are done completing your experiment, you'll move on to your conclusion questions. So we just answered number 1, what types of plants do seeds come from? Seeds come from angiosperms, and they can be either monocots or dicots. What is the purpose of a seed? The purpose of a seed is to be the baby plant, right? The embryo is the baby plant that comes from the pumpkin seed. Now, if you have parental supervision, this part is optional. You can take a pumpkin seed, preferably one of your soaked ones, not one of your dried ones, and cut carefully into it with a knife, just the very outer shell. You just want to cut through the husk. If you look inside, so these are some more pumpkin plants. You can see the green vines and the beautiful blooms of the angiosperm. This is the husk of the seed. And if you were able to cut through the husk, the seed coat, inside you will find the cotyledon as well, and they are attached by the little seed embryo down at the bottom. So if you look very carefully, you should be able to see the embryo of your pumpkin seed, right? Each one of those pumpkin seeds can be its own little pumpkin plant, and you can grow your own pumpkins. So maybe next year, you'll want to grow your own pumpkins, okay? If you were to do this experiment again, would you change anything? Is there anything that you would recommend for somebody who was to do this experiment after you? Please make sure you fill out the conclusion questions, and remember to share your results with us in your comments below. Thank you for joining us. I hope you have a wonderful October,